Thank you very much for the invitation to come. I'm delighted to be here. And for those of you who haven't yet connected the dots between mining and water, mining is a very wet business. And we now know that mining essentially controls most regional water supplies. And we need to do a much better job for the industry of giving them better solutions to protect and steward the freshwater supplies that they use. This is also going to be a story about sulfur, one of the main biogeochemical pillars in terms of global biogeochemical cycling and planetary function. And the reason that we sort of connect the dots between mining, and particularly mining waste, and global biogeochemistry is that waste environments are growing on a massive scale across the planet. And we know very little about them. So global models, even if you're not interested in mining, understanding global biogeochemical function of our planet as we go forward is going to have to take into account what happens in these contexts. Because we can't predict what happens in a mine tailings pond based on what we know about soils or oceans. So thank you very much for the introduction, Roy. Basically, in our group, we're an applied biogeochemistry group. We work in a lot of aqueous environment and reclamation contexts. The goal is to fundamentally understand the processes that drive things like water quality or stability in a waste context so that we can help the industry develop biologically informed tools and strategies. One of the biggest risks for this industry is understanding their waste, and in particular thinking 50, 100 years out, what is that reclamation closure strategy going to be in terms of success? So everything we do basically combines molecular microbiology, advanced geochemistry, but we're always thinking of this lens of how can this inform better solutions and strategies for the resource sector. And again, so the world map of minerals is essentially the world map of mines. And of course, Canada is a massive player on the global scale. And in fact, Canadian mining companies are responsible for 40% of the exploration around the world. So it's not just that we're an important mining constituent within Canada, we play a massive role around the world. And if you throw a dart at the map of Canada, you will more than likely hit a mine, whether it's active or abandoned. And we know that, of course, this has everything to do with the Canadian Shield, right? This massive horseshoe that goes around essentially Hudson's Bay. So we know that we have a lot of ore deposits, base metal, but we also have the oil sands. So we know that resource extraction is incredibly important to our economies and increasingly a part of the discussion about environmental stewardship and what we're doing in terms of water. Again, if you're not thinking about mining on a daily basis, which I realize some people don't, you have to find it. Exploration geologists have to find those ore bodies. You have to dig it. Usually a lot of our ore bodies are increasingly deeper, so we're having to dig it out. You have to extract whatever it is that you're looking to sell, your concentrate or your element of interest, whether it's nickel, copper, rare earths, and that concentrate is what you sell. But increasingly, the waste and the water issues around mining, the scale is staggering, and it's growing on a global scale. And this is something that's less well appreciated outside of the industry. The industry itself is grappling with what do they do around the massive amounts of waste they're generating, their need for water for extraction, and then what they do with that degraded water before they return it to the receiving environment. And again, so an ore is just a mining term for any rock that contains a mineral containing enough of a percent of the element of interest that you can extract it and make money, right? So nickel, copper, gold, silver, doesn't matter. Typically, you're going to be somewhere 1 to 5%, right? We're not talking huge percentages here. So that means that anything that uses resources is going to drag around a massive amount of waste in its stream, right? And this is sort of an example that I used. A lot of people tend to wear wedding rings. You're dragging around about 20 tons of mine waste, right, for every one of those gold rings. And that, you start to think about that. Do we really need to be wearing gold rings? I mean, I think there are some very fundamental questions about which resources do we really need to be going after. But one that I think is probably more appropriate, especially for the students in the room, is to start thinking about your smartphone, right, and your computers. So smartphones require about 75 elements, and we know that essentially per billion cell phones, we're somewhere around 15,000 tons of copper. You can see all of this. And in 2007, right, the decade since the first iPhone was sold, we've sold over 7 billion smartphones, and that's only growing, right? So the need for these elements that drive our technologies is only increasing. One thing I would like to ask is please don't take pictures with your phones. That's actually plagiarism. So I'd appreciate it if you would withhold from doing that. So to get at these elements that we require for everyday modern life, the industry actually requires water. And in Canada, mines withdraw about 1 billion cubic meters of surface water that they use in their extraction processes. Most of that gets returned to the receiving environment after it's been used and treated. Global consumption is somewhere around 7 to 9 billion cubic meters per year. So a large amount of our freshwater supplies, particularly in Canada, are being used for mines. And that degradation through extraction has massive implications for environmental sort of quality down the road. And we don't have a really good handle on that. Mines in Canada are probably some of the most stringently regulated. So all of their waters are monitored. They cannot discharge if it doesn't meet regulations. And if they do have a mishap, they definitely will have to deal with that. That water, essentially, so they extract, so they've got lots of rock, they're extracting whatever it is, nickel, copper, and then essentially there's a slurry that's created of this ground up rock that has water in it and a whole bunch of chemicals, which is called tailings, this waste stream, and that has to be held and managed on site before they can discharge the water. And so if you look at whether we're looking at a metal ore, whether we're looking at oil sands, fundamentally the process is the same. You dig it, you extract, and then you create this waste stream that will be a slurry of particles, chemicals, and water. And this is just to show you, it may not come out as well with the lights here. So base mine, let's see if I can get this to work. The top, metal mining, it's very highly ground up, small particles. Any geochemist in the room know that as you grind up particles, you've got high surface area to volume. You're now putting it into water, exposure to oxygen, all kinds of opportunities for reactions. Fluid fine tailings, specific to the oil sands, these are kind of interesting tailings. They don't consolidate, so they take a very long time to settle, and they tend to look sort of like a yogurty material. And if you actually drill into them, what you end up with is a liquid slurry. So these materials are also very, very reactive. And not surprisingly, I suspect for any microbiologists or biogeochemists in the room, these are full of bacteria, right, doing all kinds of things. So this is just a shot of Fort McMurray, the oil sands. So tailings facilities, and this was something I didn't really appreciate until I started hanging around with engineers a bit more, but they are in fact the largest man-made structures on Earth, and the Alberta oil sands are the largest on the planet. And the reason that we worry about this is that tailings contain a variety of contaminants, right? Beyond just metals or organics that are often used in flotation or extraction. They can have constituents that cause acidity. So they're a serious environmental threat. So mines have to hold those tailings. They have to treat the water before they can discharge it. They're often using and generating many cubic meters of tailings per day. So they are limited by the size of their retention pond. So they have to be efficient in terms of treating that water so they can release it. Otherwise, they have to stop processing. And a lot of mines in Canada will run 24/7. If they're shut down for a day, they're losing anywhere from $1 million to $10 million a day in operations, which they don't like to do. So the water is a massive piece of this puzzle, and understanding when their water quality starts to go south and they've got a problem with discharge is a really big issue for them. Mines are becoming bigger because we're seeing that essentially all of the higher grade ores have already been discovered. So what's left are lower grade ores, so lower percentages of that metal or element that you wish to extract, so higher amounts of waste. So we've got larger volumes of waste. Tailings facilities, which are often called tailings dams, tailings dikes, tailings ponds, oxidation reservoirs, depends which mine you go to, they have a different name, but the retention pond, where you put that tailings waste, they're getting larger. And oftentimes what they were originally built for, they've had to go back and re-engineer and build higher. And as we know, that hasn't always had great success. They've had a couple of large geotechnical failures in the last 10 years, particularly associated with mining. And so we're seeing that the global annual tailings production is somewhere around 7 billion tons per year, and that's only going north, right? So that's increasing on a global scale. And this is something that I often say to our mining partners, often engineers. If there are engineers in the room, I apologize, but I'm just going to say it. Oftentimes, engineers are a little bit like the Flat Earth Society when it comes to biology. They really don't want to hear that they need to think about microbiology. And I understand why, right? If we have a geochemical model, you plug in your values, it predicts what's going to happen, it's great. If we then have to add in biology, it's like, well, which bug and what do I need to know? What's my factor to model what those bugs are doing? It's much more complex. But we know that tailings are biogeochemically reactive. And this is just it's not showing up really well with the lights, but this is a tailings particle. So it's an iron sulfide particle. You might be able to see some of the blue and the red on that. Just basically what we do is we interrogate these particles with probes that are looking for DNA. And if there's bacterial DNA, those particles light up with the blue. We can look for specific kinds of bacteria. And all of this is to show that those particles, which a lot of the engineers managing mines, and particularly tailings ponds, think about only in terms of geotech, right? What's the size, what's the strength, what's the permeability, is that they actually have a living coating on those particles that are controlling the reactions that happen between that particle and the water. And they're often important, as I tell them. If your models were predicting successfully all the time, you wouldn't be hiring me to do research, and you wouldn't need to worry about this. But the models fail in their predictions of what their water quality is doing, so we know that biology is often very important to understand. And so we know that also, this is one of the sort of profoundly exciting things and somewhat challenging things about microbes, is that in the environment, they do geochemical reactions in a way that's driven by life. They have to do it under the constraints of thermodynamics, but they're very smart. So they can get together and work as partners under conditions that we would predict abiotically shouldn't drive a reaction. But the microbes have figured out a way to do this. And so this is challenging because it means that if you're predicting things based on just straight abiotic reactions and understandings of what control them, you probably aren't capturing where and when and how bacteria are driving things. So we have a whole new layer of sort of ecological biogeochemistry that we really need to understand. And this was one of our early studies where we were looking at bacteria that were essentially driving sulfur cycling in aerobic conditions. And we had the presence of both reduced sulfur and sulfate under oxygenated conditions, which makes no sense if you're thinking abiotically, but it makes all kinds of sense if you're thinking from a microbiological perspective. Bugs make their own little geochemical chambers that control geochemical conditions at the microscale. So sulfur, very interesting biogeochemically, very important from a mining perspective. Most of the world's metals come from sulfide minerals. So mining activities for any of these base metals is going to accelerate sulfur biogeochemical cycling. And if you think about sulfides, we're bringing them up from underground. You're now exposing them to water and to oxygen. You absolutely have all the conditions for geochemical reactions to occur. All the energetics, all the thermodynamic gradients, those conditions are right for something to happen and for energy to be had. And if there's one thing we know about microbes is that they're extremely adept at figuring out ways to get energy wherever there's an opportunity to do that. And of course, this is essentially a paper that came out over 10 years ago now. Paul Falkowski's thinking about planetary biogeochemical cycling. And I remember reading this paper because one of the things that it sort of was very profound for me personally, and I think the way my group works, is that we've realized we often get called in because there's a sulfur problem, or maybe there's a metal problem, or there's an iron problem. But what you realize when you look at a figure like this, which is just essentially saying that essentially the planet is no different than us, right? We work on electrons. The planet is essentially cycling electrons around to drive energy, and it's life that's doing that for the large part. But if I'm thinking about a problem that's to do with sulfur, it's more than likely linked to the cogs of carbon, to iron, to nitrogen. And so if we want to understand the context of these problems in mining context so we can figure them out and hopefully fix them, we have to think about the whole geochemical network. And so this is essentially the blueprint for how we think about interrogating and trying to understand what could be a problem in any system. I've been lecturing a lot this term, so my voice tends to sort of go about 20 minutes in. So the other piece to sort of connect to mining, because I'm often asked about mining and microbes, what's going on there, right? So in fact, if you think about it from a microbiological or particularly a biogeochemical view, so the planet formed 4.6 billion years ago. We know that life formed maybe 3.6, 3.8 billion years ago. Oxygen didn't appear until around 2.3 billion years ago, right? So there's a part where the earliest metabolisms of life were definitely not oxic, right? The earliest metabolisms are iron and sulfur. So again, if you think about it, we've got lots of these sulfide ores that we're extracting, we're bringing them up, we're crushing them up, we've put them in water, we've exposed them to oxygen. We've got bacteria that are very happy to live in these kinds of materials. Everything is there for rapid biogeochemical reactions to occur. And of course, probably one of the poster child for microbiology and mining, which is one that they don't really like, of course, is acid mine drainage, right? So this acidic, metal-laden discharge that has been known for over 100 years. And basically, waste rock, typically, which is the most well-studied, a lot of these sulfide ores, there's a lot of iron in there. As that sulfur and subsequently the iron is oxidized, you're generating acidity, leaches metals out, and you end up with this acid rock drainage, acid mine drainage. And so we've known that for a long time. And in fact, the discovery that bacteria were driving this process was made by a couple of microbiologists in the 40s. So before the structure of DNA was actually delineated in the 50s, right? So this was already identified as a microbial process. Interestingly enough, we still haven't solved that problem, right? So 70% of the world's mines still face challenges around ARD. And it is the #1 global mining issue around the world. Right? And all of these mines, so whether we look at Iron Mountain, the EPA Superfund site, the Richmond mine, whether we look in Sudbury, one of the mining partners we work with, Rio Tinto, one of the oldest mining communities in Spain, they all have the same fundamental characteristics, which is good, right? Because then it might be sort of some reactions and processes that transcend more than just one mine. And if you do Google ARD or AMD, this is the reaction that always comes up, this composite reaction for pyrite oxidation, right, that leads to FeOH3, yellow boy, and basically sulfuric acid. But one of the challenges that we've seen is that almost all of the work that's done on geomicrobiology of mining is really based on waste rock. And so our understanding of the microbes that might occur in mining waters is acidophilic sulfur oxidizers. And a lot of mining water is not acidophilic, and it's not just from waste rock. And so there's a limitation because where mines are trying to apply microbiology, they're doing it based sort of on ARD and then thinking they can use that to predict and understand what's going on in their tailings ponds. And there's a bit of a disconnect there. Because mining wastewater, so this is a tailings discharge that's coming into a mine tailings pond up in northern Ontario. You can see that discharge is a slurry of water and particles, but we will see in our tailings reservoirs that they can be anywhere from less than 3, but typically much higher pHs, and way more geochemically diverse than what you might see in waste rock. And anybody who's thinking about geochemistry or biogeochemistry knows that if you're seeing a pH range of 3 to 11, your expectation of what reactions might occur and what microbes might be there are going to be quite different. We know that that's often an important controlling factor. And so we still, though, really see these mining wastewaters as black boxes. And so for mines, when there starts to be a biological problem, they actually have no way of knowing what the processes are, and so therefore what would be an indicator that would tell them ahead of time that this process could be happening. And so what starts to happen is they start to see impacts in the receiving environment: toxicity, acidity, contamination, oxygen consumption, fish kills. So they really have a challenge, because these impacts are often difficult to remediate once they start, and they often, essentially, once they tip over, you are then treating in perpetuity, and that starts to be very costly. But we don't yet have a good understanding of who the bugs might be in these systems, what controls them, what would be indicators, are there opportunities around treatment to use these endemic organisms? And this is a risk. So one of the things that you hear a lot in the industry is this idea around risk and liability, the need for innovation. And really what that speaks to is that there's a knowledge gap, right? That we actually don't understand. And that knowledge gap essentially means that you've got risk. And as the environmental side of mining grows bigger, it's a bigger liability, it's a bigger impact for social license in order to get mines to function, because the worry is that even if you're managing this 10, 20 years out, your closure plan may not actually be very sound. And so there's more interest in understanding this now. So we started this program about 4 years ago now, and we've been sort of working to build a Genome Canada. This is the first mining project that was funded by Genome Canada, and I think it really reflects, now we're at a point with the industry where they, first of all, some of them understand that metagenomics could be a really important piece of the puzzle that they're currently lacking in terms of understanding what's going on in their systems. So we're working with multiple mines. We're working with the Ontario Mining Association, but also the Mining Association of Canada. We're also working with consulting companies, and it's an international research team. So we have colleagues at CSIRO in Australia, but as well at UC Berkeley, that are helping with this project. And so it's a large team, across multiple universities and across multiple partners in terms of industry. But the goal of this is really very fundamental, is to understand what are the sulfur compounds that occur in these tailings ponds, what are the microbes that occur in these systems, what are the new lessons we can take from this that could actually help the industry develop biological monitoring tools, management, and potentially treatment. And at the very least, steward their water that they're discharging back to the receiving environment in a much better way. And again, this is one of the sort of pieces to sort of, if you are familiar with waste rock and you work in that area, right? Obviously you think about waste rock, you're thinking about iron and sulfur oxidation. But in a tailings pond, what often happens in the tailings stream itself, particularly from sulfide ores, is that you actually generate intermediate sulfur oxidation compounds, which in Canada are often referred to as thiosalts. So things like thiosulfate, sulfite, tetrathionate, these SNOx2-. The importance of this is that these are intermediate oxidation state sulfur compounds, which can be variably oxidized or reduced. Right? So it means that we're adding to the McDonald's buffet for the bacteria in these tailings ponds. And so in a tailings pond, they can have waste rock runoff, but they will also have this tailings discharge, which has a wide variety of sulfur, as well as metals, organics, all kinds of things. And they need to treat that before they discharge. And of course, in our view, what's going to happen in that tailings pond is that what comes in from the tailings discharge is going to be recycled. It's going to be altered. It's going to be essentially cycled by microbes. So what you predict coming out of your pipe is not what you're going to see in the pond itself. And the alteration of those sulfur compounds is important because some of these sulfur oxidation intermediates are recalcitrant to current treatment. So they pass through, which means that they can then cause issues in the receiving environment. And this is not well constrained. Incidentally, I suspect anyone here who's working with sulfur knows that this is quite challenging chemically. We don't know all the sulfur species that can occur. We don't have good analytical methods for all the sulfur species that can occur. So there's a lot of work that needs to be done on sulfur itself beyond just the microbes. And so this was kind of our starting point, was to start thinking about, okay, if we think about all the sulfur species we at least know, and we have some ability to sort of measure, we kind of can think about that then in terms of, okay, here are the thiosalt species that the mines are monitoring because they think that's what comes out of their tailings pipe, That's going to be the major source of sulfur in their system. That's what they monitor, thinking that that will constrain any impacts on the environment. What we also know is that microbes, though, of course, are not just oxidizing to sulfate, and they're certainly not just doing it using oxygen. They will use a variety of electron acceptors. None of those reactions are well constrained, so they're not in models of management. Mines work on water covers to limit oxygen because they think that's how they're going to prevent sulfur oxidation. So we know that there's a much wider complexity of how microbes might be processing sulfur, and that there's probably a wider, more dynamic complexity of sulfur species because they're being recycled and altered by these microbial processes. But right now, it's a big black box. And this, as I said to my engineers at our sites, I guarantee you that your mill effluent thiosalts number is not going to be the sulfur number we're going to see in your ponds. Right? And in fact, I'm going to be getting a few bottles of wine based on this bet. So the first part of this project, and this is the first project worldwide that is systemically looking across tailings ponds at the geochemistry, the microbiology over seasonal timescales, right, to establish a baseline. So we're combining the geochemistry, molecular microbiology, and looking at those metabolic repertoires so we can start to develop ideas about what do you need to measure, what should you monitor, what could we think about in terms of managing and treating these waters. And of course, a big part of this, and I was saying one of the pieces of Genome Canada is that there's a piece that sort of links to translation. And so one of the things we're doing in this project is not just all of this applied research. We actually have Advisian, which is a consulting company, working on developing a decision-making risk-based framework for our companies to use this information. So they can make decisions at site or at headquarters about closure based on all of this science. Because it's anybody who works with industry knows they don't really want to know about all the different bugs in your system. What they want to know is, how do I use this information to do better at site or to make a better, more robust strategy for closure? And so that decision-making piece is really important. Okay, so our strategy is pretty much the same as I suspect what a lot of you guys are doing if you're sort of working in real-world environments, whether it's mining or the Great Lakes. So we're looking cross seasons, cross mines, which represent widely varying physicochemical, geochemical parameters, which presumably would also mean we might see differences in the kinds of processes that happen and or the microbes that are driving them. The way we do this, and we've been doing this in my group for a long time, so we're pretty good at it. We do physicochemistry, so we're looking in through the water cover, so we look at through depth. We use things like pH and oxygen and temperature to decide where we're going to sample. We take samples for fairly comprehensive geochemical analysis, particularly sulfur speciation. We're also taking samples for 16S metagenomic, so we can sort of look at who's there, but also what are their genomic repertoires for functionality. We're also creating enrichments of all of these samples. So we have a biolibrary that we're playing with so we can see under what conditions do these particular sulfur oxidizers drive sulfur reactions and what are the outputs. So it's a large, integrated, multidisciplinary project. And this essentially was how we set up the project. We're basically at year 2, so we've got a lot of samples. We're sort of established the baseline across our 4 mines. We're moving into test ponds this summer where we start to test out some of the ideas that we have in terms of what could be important indicators for monitoring, but also potential treatment. And in terms of what we're delivering to our industry partners, and the reason I put this up is that a lot of times when people hear what we do, which is basic research, really. We're in the discovery phase. These are important deliverables to our partners. We're not delivering a widget. We're delivering new knowledge, right? A biocollection of uniquely adapted sulfur oxidizers under a wide variety of conditions that could be applied at other mines. We're giving them bioindicators that can be used for monitoring. This idea of biological standards that can be essentially integrated to current practice and operation at mines. The geochemical model is vastly important. So even just a better model of sulfur speciation in these waters is incredibly important. So that's a big piece of what we're doing. And so this idea that only engineers, and particularly engineers that are developing pumps or treatment, are what industry is looking for is not true at all. Basic new knowledge is going to be really important to innovate for sustainability for this industry. Okay, so just to sort of give you a sense of some of the results we found. So we're working with four mines, 3 companies. So 2 of Hud Bay properties. So the 777 mine in Flin Flon, as well as Anderson mine in Snow Lake, as well as Sudbury INO up in Sudbury, and Rambler, which is in Baie Verte, Newfoundland. And so these guys have essentially been a driving force for this project. They're highly involved in what we do, very invested in not only helping us collect samples, but in terms of identifying and determining what are important results and how we sort of shape the project going forward. And just to give you an overview, the great thing about Google, right, you can go on and sort of see all of these mines. And all mines typically differ in their water systems. So this is the 777 mine in Flin Flon. Often mines are essentially mining and extracting more than one element. All the yellow stars give you the points where we've been sampling. So we're sampling at different points in their mine water system, as well as in the receiving environment, i.e. what gets discharged off-site. And so we're really trying to understand throughout these mine water systems what's going on. The Anderson mine is a simpler mine, smaller, right? So we essentially have one sort of tailings pond that we're sampling, and then Anderson Bay, which is the receiving environment. The Ming mine in Baie Verte, this was an old abandoned mine. They've reopened it. Ore levels are now economically viable, so they're remining this. There are multiple sites within this. What's interesting is that there's an older legacy tailings pond on the property, but there's a new tailings pond that they're building. So this is another set of questions that we're interested in terms of operational lifetime of mine sites. How does that influence not just maybe the geochemistry, but more importantly the microbiology that we see in a new tailings pond? Do you see the same bugs that we might see in one that's 40 or 50 years old? Because mines recirculate a lot of their water, right? So that absolutely opens the door for selection and adaptation of microbes to those systems over time. So there's a lot of very interesting questions that we're starting to go into. And finally, Sudbury. So INO is probably a mine. I've been working at Sudbury INO since I started as a prof in 2000, when it was then Falconbridge. So we've been going to this site for 17 years, and it's a fantastic mine to work at. We've got a number of sites. It's a very complex water system. We've got a number of sampling sites that we're working at. Most of our mines do not have a problem with sulfur. So they're willing to be in this project because they know that we need to establish a baseline to help those mines in the future that could have a problem. So to date, we've got 240 samples that we've collected for comprehensive geochemistry, microbes, 16S metagenomics. And so we've got a really nice set of samples that my guys are chained to in the lab processing, so that we actually have a better understanding of how seasonally does this geochemistry differ over time in these tailings ponds in the receiving environment, and more importantly, who's there and what are they doing. We're sort of in this sort of region right now. As I said, where we're going to this summer is test pond scale, so we're working at the 500 to 1,000 liter capacity tanks, and then we'll be moving from that to 50,000 liter tanks to really scale up some of the experiments we're proposing. Okay, let's look at a few results. So one of the big challenges in terms of the sulfur itself is, and one of the sort of the hypotheses we posed to our partners was that we thought one of the reasons they're having problems is that they don't actually have very good techniques right now for capturing how much sulfur is in their system. And so the first thing we wanted to do was to essentially come up with a strategy that would do a mass balance, which seems like an obvious first step to anyone who's a geochemist or a biogeochemist, but is not typically done. And in fact, is not typically done in studies of natural systems. People might measure sulfate and sulfide, but that's usually about it. So one of the first things we wanted to understand was essentially how much sulfur is in your system, and secondly, how much of that sulfur is in intermediate oxidation compounds. So like most probably of the geochemists in the room, we measure sulfate and sulfide very easily in the field by spectrophotometry. We collect samples, bring them back to the lab to do oxidation intermediate speciation by HPLC. But what we're also doing is total sulfur. And you can do this, and I would recommend this, on unfiltered and filtered samples. So you can collect, essentially, if you've got colloidal, elemental, or particulate sulfur, this allows you to distinguish that. But by doing the total sulfur, what we're able to do, because as I mentioned earlier, we don't have a good sense of all the sulfur compounds that can occur, and even for the ones that we do know can occur, we don't always have a technique to get at it. So at least if you do a total sulfur, you've got a mass balance. If we subtract all of our measured sulfur species from that total sulfur, it tells us there's a pool of sulfur that you haven't actually accounted for in your analyses. And, without sort of giving away any surprises, we see this in every sample we get. So it tells us that mines are underreporting how much sulfur is in their system. So the current assumption is they measure sulfate. Almost all mines do this. They will send out samples, typically, unless they've got a problem, in which case they might do it in-house. They will do a thiosalts analysis. This is quite hard to figure out what companies are doing, but our understanding is they're either measuring thiosulfate itself, or they're doing a technique that assumes that they've got thiosulfate and tetrathionate. And they basically do an acid digestion and figure out some value based on that. But what we're seeing is that essentially, so this is what they assume. They measure sulfate. They get a measurement for thiosalts. They've measured all the reactive sulfur they need to worry about. But what we're seeing is that, in fact, that's not true, and that even when you measure more sulfur species, we still have this unresolved sulfur pool. And it's quite significant across all of our mine sites. And so this tells us that, and it's because it's a lower oxidation state than sulfate, it can take part in sulfur biogeochemical cycling, and potentially it can move into the receiving environment. So this was one of the most important take-homes for our partners from year one, was that, in fact, they were at risk because they didn't actually have a good measure for all the sulfur that could potentially be a problem. And what we also saw, so these are just some of those numbers, and sorry, the colors changed. Here it's yellow, this unresolved sulfur pool that we get by mass balance. So this is looking across tailings ponds, but we're also seeing it showing up in the receiving environment. And that's the real issue for them, because it's making it out through treatment. There it can potentially be biogeochemically cycled, oxidized, create acidity, create toxicity, and that's where they start being in the regulatory framework where it becomes a problem. So we're now working much more on what these sulfur species are and thinking about treatment to help them with this. Okay, so who's there? And this has actually been very exciting. Obviously, I don't have all the results yet of particularly the 240 samples for metagenome analyses, but we have quite a lot of the 16S, and we have some of the metagenome results that I can talk about. So all of our metagenome bioinformatics is done by Jill Banfield's group at UC Berkeley. What's interesting about Jill's pipeline, the GGK-based pipeline, is it reconstructs genomes de novo, right? So you're not comparing against databases, you're creating those genomes essentially from the sequence data, and so we can then look at those capacities. You're not reliant on a database. So at U of T, we basically do everything from the environment to getting the sequence data. Jill's group downloads the sequence data and then does all the heavy lifting with the bioinformatics. And then we start looking at essentially what are the functional capabilities of those organisms that we're seeing in our samples. This is just an example for anyone who's interested in the bioinformatics piece of how Jill's group works on things. So check of binning, they're looking for those 51 single copy genes. So the way this is organized is these are the most abundant organisms in your sample, and then across each row is essentially the genes of identity and genes of function for those organisms, right? So you can read across and you can kind of see for each organism, does it have those genes? And that will give you a sense of what are its metabolic capabilities. And these 51 single copy genes is a way for them to see how good have they been at binning all of that sequence data to get that genome. And I think, as most people know, Jill's group is pretty good at what they do. And so of the initial set of samples we sent them, they had 276 high-quality genomes, which was pretty good. And so we know that we've got sort of a good sort of sampling, analysis, extraction, sequence data. We've got a pretty good pipeline going. They'd always like us to get more. So we're working on sort of filtering larger volumes and getting sort of more sequence data for them. But what comes out of it, and this is not one of those talks where I'm going to tell you all the names of various bugs. It's not usually very helpful, right? Unless those bugs are important to something. But one of the things that we're seeing, and I think this is quite common as well in our, a lot of the work we're doing in the oil sands, is that the proteobacteria are very common in these systems, right? In mining systems in particular. Alphas and gammas in particular are often what we see. And then, we have some other minor phyla, but you can see there's a large dominance in these samples in terms of proteos. What we also see, and this is the kind of thing that makes me happy, if we cluster those samples, just looking at the microbial communities, they're not random, right? They cluster by site, which is another way of saying they cluster by physicochemistry and geochemistry, that the communities that we're seeing in different contexts are there because the geochemistry is likely different, right? And that's important if we're trying to link, those microbes are important because they're doing something. I'd like to know that there's some consistency to who's where, right? And that's one of the things we want to tease out. But there's a very clear sort of ordination of community structure based on these different types of sites, which differ in their geochemistry. And again, sulfur is interesting / challenging, right? So not just having to think about the different kinds of sulfur compounds that you might have in water, so different oxidation state sulfur intermediates, but we also have to think about all the potential genes that can be involved in essentially catalyzing sulfur reactions. And not all of these are necessarily known yet either, right? But there's quite a lot of information on much more studied organisms, particularly acidothiobacillus, right? Very large rock stars of acid rock drainage. But we have quite a lot of information about sort of what we think are likely pathways and the genes involved in catalyzing sulfur reactions. And so Jill's group has been using this essentially to start thinking about how we can map essentially what processes are happening. And one of the first things that we saw, and I'm certainly not a bioinformaticist, would never actually try to pretend that I was a microbiologist, but one of the first things that Jill sort of said when she was looking at our data was that there was incredibly high copies of particularly SOX complex genes in all of our samples, which is very consistent with the idea. Of course, metagenomics, genomics talks about the potential for certain metabolic functions to happen. You really need to be looking at expression before you can say that activity is happening. But the high copies of these sulfur genes is very consistent with the idea that you do actually have very high sulfur metabolic activity in these waters. Makes complete sense. There's lots of sulfur in these systems, anywhere from 2 millimolar to 19 millimolar, right? It's a lot of sulfur. What we also see, which is also interesting, again, getting back to this idea that microbes are catalyzing sulfur oxidation or disproportionation beyond just coupled to oxygen, is that a lot of our organisms that are capable of sulfur oxidation are able to also do nitrate reduction. And there's a lot of, I suspect a lot of you guys know, there's a lot of emerging evidence that sulfur in particular is being coupled to nitrate reduction, to sunlight, to iron 3. So this idea that you're going to manage sulfur only through oxygen doesn't make sense if we're starting to see that there's a much wider biogeochemical array of how bugs could be doing this. And it's very consistent across all our samples. And so, kind of getting back to this idea that if we really want to understand the outcomes or the outputs of a tailings pond, we actually have to think about this much bigger biogeochemical connection amongst these elements. And in particular, the nitrite one is important in mining because you often see quite high concentrations of nitrogen species because of blasting, right? The explosive chemicals. And they're often, particularly nitrate, will be higher in concentration than oxygen. So if you're managing to prevent sulfur oxidation, because you think your oxygen concentrations are low, but these guys are capable of using nitrate, and nitrate concentrations are high, you really haven't captured the cycling that's going on in your system. So we're sort of working on quite a few experiments right now to better constrain some of this. Okay, so, and this was a question that we got when we were starting to set up this project. In fact, I got a couple of people sort of saying, we don't need to look at tailings ponds. We've been working on waste rock for ages. We know it's acidothiobacillus, we're good to go. And I said, well, I'm not sure that we are. I think we really should sort of examine, are the bugs that are doing sulfur metabolism in wastewaters the same as what we see in acid rock drainage? And even as a geochemist, there's all kinds of reasons that would suggest they aren't going to be the same. But this was a very fundamental question that really hadn't been answered, which sort of shows you how much we need to do in these contexts to really understand who's there, what are the role of microbes, and what are the opportunities to use them. And so the way we've set up our enrichment, so enrichments, again, if you're not a microbiologist, right, we get a sample of water or soil, we know that there are organisms within that sample that are capable of metabolizing sulfur. We'd like to get them out, and we'd like to see what they do. So we give them essentially a media that's got food in there that we think will select for those guys that are capable of oxidizing sulfur. And so what we've been doing is we've been playing games with which sulfur oxidation intermediates we give them, the conditions under which we grow them, because it's not just a question, I think, of giving them some sulfur. I think we're seeing ecological adaptation to pH, to temperature, to oxygen, to light, so we're playing all kinds of games. And we'd like to know, if I give water samples from 4 different mines collected the same month, if I give them the same media, will I get the same bugs? i.e., is it dependent on the geochemistry of the growth media, or will they be different bugs, but doing the same thing? i.e., it's dependent on the mine which they came from. And I think the answer is it's a little bit a combination of the two. But what we're seeing is it's not all the same. So I'm going to talk just today about our first publication on this, which is pretty exciting. We have many more enrichments. We have a lot more data on this. But this is actually a pretty exciting story because we really, my supposition would be if I got water samples from 4 different mines, quite different in their geochemistries, quite different in pH in particular, you wouldn't expect to see the same bugs come out. And so we looked at 9 samples across all 4 of our mines that we collected between September and November 2017. We looked at the reservoirs, we looked at some inputs, and we looked at the receiving environment. And we took all of those samples and we gave them essentially all the same sulfur media to see who would grow when we looked for sulfur oxidizing bugs. But if we looked at the parent waters from all of these different mines, what this essentially shows you is that they cluster by site, right? And that's not surprising. So you can see there's variability, but essentially those mine waters cluster by site. So it would suggest the mines, there's some variability that's site dependent. And so this is just the 16S data, sort of looking, so from mine 1 to mine 4, right? These are the tailings reservoirs or ponds themselves. We have an input here and we have a receiving environment. And then, so I promised you I wouldn't talk about names, but the one to watch is this yellow one, right? Halothiobacillaceae, this family here. And so this is not surprising. I suspect many of you guys have seen many data like this where the pie diagrams are all different, right? For different samples. They're different microbial communities. We would expect that. But what was really interesting is when we looked at the enrichments from these samples, is that the vast majority of them, and in fact we have a couple that are 97 to 100% Halothiobacillaceae, which is really quite remarkable to have enrichments that converge from very different mine sites that converge on this dominance of this one family of organisms. These ones here, slightly different, and I'll talk about that mine in a minute. What's also interesting, if you look at this particular receiving environment and this tailings reservoir parent water, those 2 sites, when we sampled them, were acidic. And so that's also something that's interesting I'm going to come back to. So Halothiobacillaceae, and I'm mixing the 16S and the metagenome data, which is also great because it kind of converges on the same story, but we get more insight from the metagenome data. So it's found in all the mines, this Halothiobacillaceae. It should be, if we're able to grow it, it needs to have been there in the parent water. They all have genes for sulfur oxidation, so consistent with our ability to grow them under sulfur oxidizing conditions, they're the ones who come out. As you can also see, they also mostly all have genes for nitrate reduction, which means that they could couple, theoretically, sulfur oxidation to nitrate reduction rather than just oxygen. And we know they're doing this. The other thing that was very interesting, so this is just looking at a phylogenetic tree, and this is based on our 16S data, but this is looking at the mine parent waters and then our enrichment Halothiobacillaceae. So all of the colored ones are ones from our samples. And then we've compared them to ones that are actually in known databases. What's important about this, two things, I think. One is that these are similar, but not really, to ones that are in the database. So we know that mining environments, I really think there is a mining microbiome, and it's distinctly different than what you might see in soils or oceans, right? So we're seeing all kinds of new Halothiobacillaceae that haven't been found before. And most importantly, so here are Acidothiobacillus, right? The huge rock stars of ARD. You can see that they are not particularly similar to Halothiobacillaceae, right? So important organisms that are driving sulfur oxidation in mining wastewaters, tailings ponds, are not the ones that we find in waste rock. We're delving into their genome because, okay, you know, what are the differences there and what are the implications of that? Obviously, we want to know. But Halothiobacillaceae has been known to do sulfur oxidation, but they certainly have been under everybody's radar. No one knew that this was an organism that could be very important in mining wastewaters. The fact that we're converging on it in a number of samples suggests we could have a broadly important organism across a wide variety of mines. Okay, so if we think about, you know, how bugs might be processing sulfur, and it's important because, so bugs, rather than going from, you know, low, highly reduced sulfur to sulfate, are not going to do this all in one shot, right? They're going to control those electrons because they get more energy. So it's typically 1 to 2 electrons at a time. And we see a lot of them are able to recycle, to disproportionate sulfur, so they produce one oxidized and one reduced. So we have a lot of cycling of sulfur. The importance of that is it decouples sulfate to acid ratios. And oftentimes mines will measure pH or sulfate, assuming that it's a linear oxidation, and usually that's sort of 1-to-1 sulfate to H+, and they predict what's happening with their sulfur. If disproportionation's going on, your sulfate to acid ratios are decoupled completely, and you're not accurately constraining what's happening with your sulfur. And what we see is that, okay, we have all these Halothiobacillaceae, does it matter? So if we look at essentially the composition in our enrichments of all of these different genera of Halothiobacillaceae, we look at what they're doing in terms of acid to sulfate, it differs quite a lot. So we know that this starts to give us a sense that actually we can now drill down a little bit and understand if you have, you know, 2, 6, and 7, you might get this, and we'd like to know what are the conditions that drive that and what are the outcomes. So we're spending a lot of time delving into this. I think these are the kinds of things that would make really good biological indicators. And what we see, and this is going back again to sort of looking at the Halothiobacillaceae, what's very interesting as well is that the ones that dominate in the enrichments are not necessarily the ones that dominated in the parent waters. Right? So that also could be a really important indicator of when things start to go south in terms of generating acidity. If we start to understand what are the tipping points, what are the triggers that change which of the Halothiobacillaceae are present in your samples. And I think this is the kind of important information we can get out by essentially coalescing geochemical data with 16S data, but more importantly with metagenome data in terms of understanding what processes are happening and what are indicators of that. So, you know, when we sort of looked at this, as I said, one of the things, because obviously anyone who works in enrichments, you know, there's limitations to what you get to grow in the lab under highly controlled, very artificial conditions. Essentially, does that represent? And this was the point that we see them dominate in these environments where the pH had already gone less than 4. So this was a really nice way to go back to our real sites and show, actually, very consistent with the idea that these guys are important for starting that sulfur oxidation train that leads to acidity. They're present in the environment. So this is my last slide. And really, it was just, this is a figure that we sort of put together for the front. This just came out in Frontiers. This paper was to point out that, you know, the understanding of microbiology has really been on waste rock, and this idea that Acidothiobacillaceae are the kings of or queens of iron and sulfur oxidation. But we're seeing in tailings ponds where we have a much more complex geochemistry that we are seeing that we might also have this important group of organisms that could be important for initiating these sulfur reactions. What we've seen in subsequent work is that we actually see that Thiomonas takes over from the Halothiobacilliaceae. So we're starting to see some pH progressions, which we can link to ecology. And all of this is to say that, there's obviously a lot more we need to do, but I think this kind of integrated platform is going to be critical for opening up new doors for the mining industry. So with that, I'd like to say thank you and really thank my group, because they're the ones that are kind of doing all of this work, and thank you for your attention.